This is an overview of the U.S. Environmental Protection Agency's Cryptosporidium and Giardia detection methods. The procedures focus on concentrating a volume of water to capture Cryptosporidium and Giardia and seeing these disease-causing organisms through a microscope. Cryptosporidium and Giardia detection begins with a 10-liter water sample that is filtered to concentrate the organisms and other suspended particles. The particles are then rinsed from the filter and further concentrated into a pellet by centrifugation. Cryptosporidium and Giardia are isolated from the extraneous debris by a process known as immunomagnetic separation. The final steps are to stain these organisms bright green for examination by a skilled microscopist. We begin with the filtration of a water sample. The date and time of sample collection are recorded to document timely analysis before the microbes degrade. During filtration, the suspended particles within the sample are captured on a filter. Clean tubing is attached between the sample, filter, pump, and a waste container. Once the filter is attached into the system, the pump is turned on to flow at a rate of 2 liters or less per minute. The container is emptied and the pump is turned off before the fluid drains from the filter capsule. The container is rinsed with reagent water. The rinsed water is pumped through the filter. Then the filter capsule is capped for transport. Before 96 hours has elapsed, the filter processing is started and the date and time are recorded. 125 milliliters of 5% sodium hexametaphosphate solution is added through the inlet of the filter capsule to disperse the organisms from the extraneous sample debris. The filter capsule is then clamped to a wrist shaker at maximum speed for 5 minutes. The sodium hexametaphosphate solution is pumped through the filter. The dispersant is rinsed from the filter with reagent water. Elution continues by adding 125 milliliters of buffer solution to the inlet of the filter capsule. The capsule is clamped for shaking at maximum speed for 5 minutes. The contents of the capsule are poured through the inlet into a centrifuge tube. More buffer solution is added to the filter and shaken 2 more times at maximum speed. The filter eluate is added to the centrifuge tube along with a rinse of the filter capsule. The centrifuge tubes are balanced within a half a gram of each other. They're then centrifuged at a minimum of 1500 times G for 15 minutes and allowed to coast to a stop without the use of a brake. The resulting pellet volume is compared to standards for measurement and recorded. The supernatant is aspirated until 5 milliliters of liquid remain above the pellet. The pellet is homogenized by vortex. Immunomagnetic separation is used to separate the Cryptosporidium and Giardia from the debris in the pellet using magnets housed in 2 different plastic holders, a rotating tube mixer, and an IMS kit containing buffer solutions and magnetic beads coated with a specific antibody. 1 milliliter each of buffer A and buffer B is added to a flat-sided tube. Using a pipette rinsed with buffer solution, the sample is added to the flat-sided tube. The tube is rinsed twice with reagent water. The magnetic beads are mixed, and 100 microliters of each is added to the sample. The tube is rotated for an hour, allowing the magnetic beads to bind to only the oocysts and cysts in a specific reaction. The flat-sided tube is then placed in the larger magnet housing. The tube is rocked slowly end-to-end for 2 minutes. The supernatant is poured from the tube, with the flat side up, so that the beads which are attached to the Giardia and Cryptosporidium are not disturbed. The tube is removed, and the sample is resuspended and transferred into a microcentrifuge tube. The flat-sided tube is rinsed twice. The tube is gently rocked against the magnet end-to-end for a minute. The oocysts and cysts are thus trapped onto the magnet with the magnetic beads. The supernatant is aspirated from the tube. Any remaining debris is rinsed from the beads using a PBS buffer solution. The magnet is removed, and the beads are resuspended in the solution. The magnet is replaced, and the sample is gently rocked end-to-end for 1 minute. The supernatant with the extraneous debris is aspirated from the tube. The magnet is removed, and 50 microliters of hydrochloric acid is added to the sample and mixed on a vortex to break the bonds between the beads and the oocysts and cysts. The sample is allowed to stand for 10 minutes at room temperature. Then the tube is vortexed again. The magnet is replaced to bind the beads to separate them from the Cryptosporidium and Giardia suspended in the liquid. To neutralize the acid used in the previous step, 5 microliters of sodium hydroxide is placed on a microscope slide. Then, the supernate containing the organisms is transferred onto the slide. The date and time is recorded before the sample is dried on the slide. The sample is stained within 3 days of slide application. The first stain, called DAPI, is used to highlight the DNA in the organisms. After a minute, the stain is diluted with wash buffer. The well slide is tilted, and the excess DAPI is removed from the slide. FITSI stain is applied to the sample to specifically highlight the walls of the oocysts and cysts. The slide is placed in a humid chamber for 25 minutes. Then the FITSI is rinsed from the slide. Mounting medium and a cover slip are applied to the slide. The slides must be examined before the 7-day holding time expires. To begin the microscopy process, the microscope oculars should be adjusted for the vision of the microscopist. A Giardia cyst is found at 20x using FITSI and brought into focus using the coarse and fine adjustment knobs. Next, the microscope is adjusted for maximum contrast and resolution using a procedure known as Kohler illumination. The height of the condenser is adjusted into focus by the microscopist, viewing the edges of the field diaphragm. Then the analyst centers the aperture using the condenser centering screws. The aperture is open to illuminate the field of view. The Kohler illumination procedure is repeated at 100x. Once the light is aligned, the condenser is switched to the differential interference contrast, or DIC position. Notice how the images go from 2-dimensional to 3-dimensional. Every oocyst or cyst found in a water sample must be measured and characterized with FITSI, DAPI, and DIC. Cryptosporidium O-assists are brilliant green with FTSE, measure between 4 to 6 micrometers in diameter, and display unique characteristics by both DAPI and DIC. Giardia assists measure between 8 to 18 micrometers and are also brilliant green with unique characteristics. For more information on the Cryptosporidium and Giardia detection methods, please use the resources available on our website.